Welcome to Life in Biology. I'm Dr. Joel Graff, and this is a condensed lecture for my Discover Biology class at Montana Tech. It's Biology 101. This lecture goes over some basics of DNA structure and replication. So DNA, we learned earlier in our course, is a polymer. RNA is also a polymer. And polymers, poly means many and mer means part. And so the smallest piece of a polymer is the monomer, and mono is one and part. So you start stringing together monomers and you end up with several parts or a few parts, and so that's an oligomer, and you keep adding to your growing chain, typically it's a chain, then you end up with polymers. So for the different polymers that we're going to deal with, DNA, protein, carbohydrates, we're going to break it down and look at the individual parts. So the nucleotides are the monomers. You string a couple of nucleotides together and you end up with an oligonucleotide. And then you keep adding to that and you end up with a polynucleotide, which is not very commonly used, and instead nucleic acids is more commonly used than polynucleotide. Over here I've drawn out an example of a nucleotide. This doesn't show all of the different pieces to it, particularly the NB here is a nitrogenous base. And that has quite a few atoms that are involved in that. But for this class, what we need to know is that DNA, the nitrogenous base is going to be either an adenosine A, a cytosine C, thymine T, or guanine G. Nearly the same four letters are used for RNA, but one difference you can see is where in DNA we had a thymine, in RNA we have a uracil. So A, C, U, and G are the letters. What I'm showing with this black box or outline here is I'm trying to indicate that a nucleotide has an L shape to it. Nucleotides can start with 3 phosphate groups on it, but usually 2 get cleaved off, and so when you start stringing a bunch of these nucleotides together, the shape is an L shape from this one phosphate, this sugar, and then a nitrogenous base. So that L shape is important. Again, I haven't drawn all the detail nitrogenous base, but I've also left out some stuff here on the sugar. Importantly, I want to point out that this is a ribose sugar. We have carbons 1, 2, 3, 4, and 5. The 5 carbon has a phosphate group, and we're going to come back to that. The 3 carbon has a hydroxyl group, and we're going to come back to that. The 2 carbon can either be a hydrogen or a hydroxyl. In DNA, in place of this R, you would have a hydrogen, and in RNA, in place of this R, you would have a hydroxyl. So the difference between a hydrogen and a hydroxyl is the oxygen. So RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. So that deoxy is just indicating that you don't have that oxygen here on the 2' carbon. So that's a big difference between RNA and DNA, is what is connected to the 2 carbon right there. Next, I mentioned that this was an L-shaped molecule, and so I wanted to point out that if you draw an L like this, and then you come along and you draw another L, and another L, and another L, and so on and so forth. Here I changed colors, and I'll get to that in a second, but they end up forming a string along the top, and then you get some branching points coming off like this. So this branching points here would be the nitrogenous bases, would be out here on the the branches. And then the sugar would be right where the L bends. And then the phosphate would be right here. One thing, remember we've got a phosphate there, so that's the 5' end. And then there would be a hydroxyl here. And we'll come back to that later, but that's where that hydroxyl will get kicked off when the next nucleotide gets added to this growing chain. The AGCCAT that I drew out here was just to indicate that I had drawn some color coding here. So for instance, green L's represented cytosines, red L's represented adenosines, and so on. But that's not too important. I was just trying to make the point that you could take any of those 4 nucleotides and add them to the growing chain. Okay, because drawing a bunch of L's all the time gets a little bit tedious, what you can do is you can draw a straight line to indicate the sugar phosphate, phosphate sugar backbone, and then these branching points here, the tons of blue little lines that I've drawn there are indicative of the nitrogenous bases. So that's one strand, the blue strand. The second strand, we've got a second strand here. So this is showing double-stranded nucleic acid right now. We don't know if it's DNA or RNA because we can't see what's on that 2' carbon of the sugar. But anyway, you have 2 strands. You've got the sugar phosphate backbone on this strand and the sugar phosphate backbone of this strand. They're all held together with covalent bonds, and they're covalently linked also to the nitrogenous bases here. And then if you remember, what we learned in our class earlier was that there were hydrogen bonds that connected the nitrogenous bases of this strand to the nitrogenous bases of that strand. And then in the class, this is really messy. I was just indicating that sometimes I do get messy drawing DNA, and I just draw a couple lines, and then I just do some quick bars like that. And each of those rungs in the ladder basically represents the 2 nitrogenous bases interacting between your 2 strands of DNA. Okay, anti-parallel structure. So I drew another ladder looking thing here, and what I want to point out is that this is the 5' end of the strand and this is the 3' end of the strand. On the second strand, the 5' end is at this end and the 3' end is at that end. Associated with the 5' end is a 5' phosphate group here and here, and associated with the 3' end is a hydroxyl here and here. And I'm bringing that up because we're going to be needing those hydroxyls here in a second. But the structure of DNA is considered anti-parallel, and then parallel. So parallel because this strand and this strand are running parallel to each other, and then anti because this strand goes 5' to 3' as you go left to right, and this strand goes 5' to 3' as you go right to left. Essentially what that means is if I were to draw in the L-shaped brackets here, it would look that direction for that strand, and it would be like that for the second strand. Okay. So the numbering again was based on the sugars, and that was over here. 5' phosphate, 3' hydroxyl. Sometimes I draw DNA as a ladder that's horizontal, and I'll always have 5' to 3' going left to right, and for the top strand, and 5' to 3' going right to left for the bottom strand. In the same way, there's a standard way of drawing nucleic acids. If you're going to make your ladder standing up, you want the 5' end here and the 3' end down. So it goes 5' to 3' in, let me grab my arrow, 5' to 3' in that direction, and 5' to 3' in that direction. How are you going to remember which one goes where? The 5' is in the upper left here, and the 5' is in the upper left here. So both times we have it in the upper left. Sometimes in my classes I talk about how you hang a flag on the wall. And if you hang the American flag on the wall, if you have the flag hanging downward like this, the field of blue is in the upper left. And if you have the flag hanging horizontal, so your red and white stripes are going horizontal like that, your field of blue with the stars should be in the upper left as well. So having standards for how you draw your DNA out will come in handy, and when we get to DNA replication, we'll see why. Okay, before we move on though to replication, the last part of the structure that I want to talk about is that adenosines form are always on. If you have an adenosine on one strand of your nucleic acid, and on the other strand, in the case of DNA, it would be a thymine. So A's match up with T's, and these red lines here are hydrogen bonds. So what I'm indicating is that an A is interacting with a T through 2 hydrogen bonds. C's can interact with G's, and that's with 3 hydrogen bonds. The other thing to point out is that A here and G are called purines, and so they, in the structure, if this is your DNA or your phosphate sugar backbone, we're going 5' to 3' in this direction, and 5' to 3' in this direction, right? If that's your sugars and phosphates making up the backbone of each strand, then those rungs going across, the A's and T's are going to have 2 hydrogen bonds going between them, and the C's and G's are going to have 3 hydrogen bonds going between them. And I'm not going to get into the structure of adenosine, cytosine, guanine, and thymine, but if you do Google their structure, what you're going to see is there's covalent bonds linking the sugar out to some structures out here that are also kind of circles, cyclical, and A's have 2 circles of atoms, and T's have 1 circle of atom. G's have 2 circles, and C's have 1 circle. The A and the G are purines, and so that's why they have the same number of circles. If we talk about the T and the C, those are called pyrimidines. Again, we're not going to worry too much about the names of those, but the main point I wanted to show here is that there is basically 3 circles between the strands, between the riboses. And so 1, 2, 3, 1, 2, 3. And what this does is, because those circles are roughly the same size for A's and G's, C's and T's, you're going to keep the same distance between this strand and that strand, and so that's important for making sure that those strands are parallel to each other. Okay, moving on. The enzyme that's involved in copying nucleic acids are called polymerases. Polymerases will read one strand in a nucleic acid. If they see a C on one strand, they'll put a G in on the other strand, or if they see an A in one strand, they'll put in a T in the other strand. If you're dealing with DNA, remember T's are replaced with U's for RNA, so A's and U's would match up in an RNA molecule. So this is the enzyme that creates the new strand of DNA or RNA. Because we've got DNA and RNA, we've got 4 categories of polymerases. And so if you want to describe a polymerase, because there's 4 different types of polymerases, sometimes you want to explain what polymerase you're dealing with. And so you would say either DNA or RNA dependent DNA or RNA polymerase. So DNA or RNA could fill in this blank, DNA or RNA could fill in this blank. So let's just run through those 4 possibilities. If a polymerase reads DNA and makes DNA, it is a DNA-dependent DNA polymerase. So you would write DNA-dependent DNA polymerase. Okay? But if it reads DNA and makes RNA, it would be a DNA-dependent RNA polymerase. And so you can shorten that up to a capital D for DNA, lowercase d for dependent, capital R for RNA, lowercase p for polymerase, DNA-dependent RNA polymerase. If it reads RNA and makes RNA, it's an RNA-dependent RNA polymerase. And if it reads RNA and makes DNA, it's an RNA-dependent DNA polymerase. And so those are the 4, and that will come up again in a future lecture. Okay, so let's say we're wanting to read DNA and make DNA. So this is copying DNA. Well, the way that DNA is copied is described as semi-conservative replication. You're going to copy your DNA. Semi-conservatively means it's halfway keeps the same. Okay, so I started out here drawing some double-stranded DNA, and I have 2 strands, and they're both black. The red dots here are just indicating the hydrogen bonds holding those strands together. Because the backbone and the phosphate, the sugar, and nitrogenous base are all held together with covalent bonds, and they're connected with a covalent bond to the next nucleotide that has the phosphate, the sugar, and the nitrogenous base. Those are all very strong bonds. Covalent bonds are strong. Hydrogen bonds are weaker, and so they're going to be easier to break apart. So what happens is you can come in, break those apart either with heat or with an enzyme that's able to basically plow in there and start breaking things apart. You can end up with 2 double strands of DNA, where one strand, this top strand of this DNA strand, was the top strand of this original DNA. This double-stranded DNA, the bottom strand of this DNA strand, is the bottom strand of this DNA. And then we had a polymerase acting to read the DNA and make the DNA, read the DNA and make the DNA, and that was able to give you a double-stranded DNA. So it just read the polymerase would read the A's, C's, T's, and G's on this strand, and then put in the correct Watson-Crick base to go across from it. So when an A and a T are matched up, it's called a base pair. If C's and G's are matched up, it's called a base pair. So if you say the length of a double-stranded nucleic acid, you would use the word base pair. If it was just single-stranded, you'd use the word nucleotide if you were describing its length. So polymerases read an old strand as they make a new strand. So again, halfway keeps the same. Alright, and the final topic today is jumping into the DNA replication fork. So this replication from one double-stranded DNA to 2 double strands of DNA, 2 sets of double-stranded DNA, doesn't just happen by magic. In your cells, you have to have proteins or enzymes going through and doing the reactions. And so there's quite a bit to this image. And so I'm just going to start out talking about these 2 black strands that we have here. This one and this one are the original double-stranded DNA. So we know that the top strand should go 5' to 3' as we go in that direction. And if we follow this strand out, I'll connect those, we end up at the 3' end. We know the 5' end has a phosphate, and we know the 3' end has a hydroxyl. Don't worry about any of these things I've drawn on here so far. We're just paying attention to the strand. The bottom strand will have a hydroxyl at this end because that's the 3' end. And if we follow it over to this end, we've got the 5' end with its phosphate. So it goes 5' to 3' in that direction. Okay. Let's just work on the enzymes here from left to right. We've got a couple of proteins here. We've got a gyrase and a topoisomerase, but I'm not too worried about you remembering the names of those. What they are doing is that there gets to be some stress built up in this double-stranded DNA, because remember, this is not really a ladder, it's more of that double helix, like the spiral staircase look to it. And so if you start to unwind a couple things that are wound around each other, you could build up a bunch of tension. So the gyrase and the topoisomerase, their purpose is to get rid of that stress. They do it by cutting and unwinding that DNA. Then the next enzyme we'll talk about is the helicase. So I like to draw a helicase as a triangle, because I picture the helicase moving in this direction and breaking those hydrogen bonds that are formed between those. So the helicase will move the way that we have this picture drawn. We have the helicase moving from right to left, and it's going to plow in and break apart those hydrogen bonds. We have two strands of the original DNA. We've got this strand, and we're going to call it the leading strand. And we've got this strand, and we're going to call it the lagging strand. The leading strand is easy to make double-stranded. The reason for that is that polymerases, in this case we have a DNA-dependent DNA polymerase, it depends on a 3' hydroxyl to place in the next nucleotide. So if we have a nucleotide, it's going to get, and remember that's the L-shaped thing, it's going to get inserted here, kick off that hydrogen and form a covalent bond with the carbon that was here. And then the next nucleotide comes in. So we got one nucleotide coming in and then another nucleotide coming in as we go along and move in the 5' to 3' direction. So polymerases always move 5' to 3'. So in this case, the helicase in our figure is going from right to left, and it's separating out those two strands of double-stranded DNA. And in this case, this black circle here represents my DNA-dependent DNA polymerase, and it's just going to follow right along after that helicase. And so no matter how far the helicase goes along, the polymerase will just follow right along. So the leading strand is easier to explain. The lagging strand is difficult because we need a 3' hydroxyl, and if there was no nucleic acid on this second strand, there would be no 3' hydroxyl to start building off of. There's another enzyme called a pyrimase, and it comes in and it occasionally makes stretches of RNA that match up with the DNA. And when it does that, it leaves the RNA sitting there, and this 3' hydroxyl on this green RNA stretch is going to work just fine to put in the next nucleotide of DNA, and then you start filling in your nucleotides, and the polymerase moves in this direction. So that 3' hydroxyl would get kicked off. You're building your nucleotide chain, and you've always got a 3' hydroxyl to work with. So this polymerase is going to work in a 5' to 3' direction, and as it does that, it's going to get down to this another RNA that was laid down by a pyrimase. That should say 3, and it's going to work in a 5', which is sitting there. And when it gets to that RNA, this polymerase falls off, and a separate polymerase comes into play, and it has the ability to chew up the RNA, and so the RNA will get degraded, and the second strand will be entirely of DNA. This RNA that was here served as a strand for building the polymerase and having the polymerase be able to work off of a 3' hydroxyl working in a 5' to 3' direction down here. So this is kind of messy, but hopefully by walking through it step by step that kind of made some sense. Anyway, because you have to keep doing this, lay down an RNA, build off of it, and then chew up the next RNA to fill it in. This is called the lagging strand because there can sometimes be a bit of a time lag before the RNA primer is laid down and a polymerase is able to come in and fill out that second strand. The leading strand again. All it has to do, this polymerase, all it has to do is follow the helicase as it goes in that direction. And then so that wasn't my last slide, but we just got done talking about DNA replication, and if you look at the central dogma of biology, which I always come back to in all my classes over and over and over again, DNA can be replicated to make more DNA. So we've talked about replication. DNA can be read, and then a new strand of RNA can be made, and that process is called transcription. RNA can be read by ribosomes and that made into protein, and that's a process called translation. So transcription, DNA to RNA to protein, is the flow of genetic information, and transcription and translation are the two steps. Okay, we're going to come back to central dogma of biology in the next lecture. So I just want to compare the steps of transcription to the steps of translation. If you remember back to metabolism, we kind of broke things down into groups of reactions so that it was easier to remember. So like cellular respiration, we had glycolysis, pyruvate metabolism, citric acid cycle, and electron transport chain, and then the ATP generation. Here, we're going to have 3 steps for transcription. The first step of transcription is initiation. That means recruiting in the DNA-dependent RNA polymerase. The second step is elongation. That's when the DNA-dependent RNA polymerase moves in a 5' to 3' direction along the DNA. So you have to bring the polymerase in here, and then it moves in the 5' to 3' direction here. And finally, this last step of transcription is termination, and that is when the DNA-dependent RNA polymerase releases from the DNA, and if it's releasing from the DNA, it can no longer read any template, so it stops making new RNA, and your RNA is completed at that point. Translation, that's reading RNA and making protein. Initiation is the recruitment of a ribosome to the first codon of your protein, and we haven't talked about codons yet, so that might be putting the cart before the horse, but codons, every 3 nucleotides of RNA encode for 1 amino acid. So those 3 nucleotides together, it makes the code that is encoded, or that gets made into the amino acid. The ribosome, again, it's a big complex enzyme that will do this. Second step of translation is elongation. That's when the ribosome reads the mRNA and works in a 5' to 3' direction. And we'll talk about that in the next lecture as well. Termination is when the ribosome releases from the RNA, and if the ribosome's not reading RNA anymore, it obviously stops making protein. And I'm missing the I there, and this is an E. The smartboard in the lab or the lecture hall won't let me write in the bottom right corner. So that's what's going on there. So that gives you a bit of a preview of where we're going to go in the next lecture. Anyway, Hope you enjoyed this lecture. Like and subscribe, or not.